Today we shall discuss the topic Selaginella. As all of you know, it is a very important genus in lower pteridophytes. The classification for Selaginella is Lycopsida, Ligulopsida, Selaginellales, Selaginellaceae, and of course the genus Selaginella has numerous species. Around 700 species have been reported for Selaginella, and some important species are Selaginella kraussiana, Selaginella chrysocollus, Selaginella rupestris, Selaginella lepidophylla, like this. Now coming to the habitat and the habit of the plant body, Selaginella prefers to grow in moist, rich area, and of course it is a very wild plant growing in and around this particular place, and it is very common in the tropical areas. Coming to the habit of the plant body is a sporophyte, and the sporophyte can be differentiated into root, stem, leaves, and one organ of morphological curiosity, that is rhizophore. The root is simple, it is rhizoidal. To some extent it helps in anchorage as well as absorption. The stem is almost semi-transparent, it is slender, either dichotomously or monopodially branched. It is very delicate in some species. The leaves are usually microphyllous. A microphyll is a leaf having single median unbranched vein. And in case of Selaginella, of course, we can see two different types of leaves, and the differentiation is only with respect to their size. Some of the microphylls are bigger and some of them are tinier. And the leaves are usually arranged in a spiral pattern, and they possess a very important structure on the adaxial surface, and these structures are called by the name ligules. And a ligule is nothing but a tiny needle-like projection that is found at the base on the dorsal surface of the leaf. It is believed that the ligules help in maintaining the moisture level of the leaves. I mentioned about one organ of morphological curiosity, and this organ is nothing but rhizophore. It is a peculiar organ found only in Selaginella, and the rhizophore is colorless, it is positively geotrophic, it is a root-like, it is a stem-like structure. In the meantime, it can be regarded as an organ of morphological curiosity, that is an organ sui generis. An organ sui generis is an organ which cannot be compared to any other existing organs. Now coming to the internal structure of the stem, in Selaginella the stem is very specialized because it shows polystelic protostelic condition. The stele is simple, stele is protostelic, that means the pith is absent, and numerous steles can be seen in a particular section. When the cross section of the stem is taken, sometimes as many as 16 steles can be seen. Because of this, it is referred as polystelic condition. And individual steles are haplostelic, and the steles are suspended in a lacuna, and they are suspended with the help of some organs called by the name trabeculae. And when the transverse section of the rhizophore is taken, it shows almost monarch exarch condition at the stellar region, and rest of the things are almost similar to any other cross sections. Coming to the reproduction in Selaginella, it is somewhat different because Selaginella shows heterospory and seed habit. This is one particular event witnessed in pteridophyte, and it is very peculiar and very specialized and exclusive for Selaginella. The Selaginella sporophyte reproduces by the formation of the spores, and it produces two types of spores. The two types of spores are microspores and megaspores. The microspores are produced within microsporangia, megaspores within the megasporangia. And the microsporangia arises on the microsporophyll, whereas the megasporangia arises on megasporophyll. And these micro and megasporophylls are congregated to form the reproductive organ, that is strobilus or the cone. When the LS of the cone is taken, it shows the central axis and the sporophylls on either side. And we can see the micro as well as megasporophylls arranged in various patterns. In some sections we can see the microsporophylls only at the tip, megasporophylls at the base, and sometimes we can see them arranged in an alternate pattern. Now, the megasporangium possess limited number of larger spores, and microsporangium produces numerous microspores. After their development, the microspores develop into male gametophyte, whereas the megaspores develop into female gametophyte. Now, each megaspore as well as microspores, they show their earlier development. That means the initial development of the male and female gametophyte take place even when the spores are found within the sporangia, and even when the sporangia is found attached to the cone. This is a peculiar property that is known as seed habit property. Selaginella shows heterospory, a phenomenon characterized by two different types of spores, and seed habit means the tendency of the so-called female gametophyte to remain attached to the mother plant even after the formation of the so-called young sporophyte. Though Selaginella is a lower primitive pteridophyte, it shows such an advanced character which is very much pronounced in other angiosperms. Of course, the development of the micro and mega gametophyte take place inside the respective sporangia, and later the process of fertilization sets in, and ultimately the zygotes are formed, and the zygotes develop into the young sporophyte. Hence, the speciality of Selaginella is they possess rhizophores, they show polystelic protostelic condition with trabeculae, and in life cycle they show heterospory and seed habit. Thank you.